audio?
Hi, everyone. This is David Spiegel. I am speaking to you from my office in New Haven, Connecticut, regretting very much that I was not able to make it to the conference this year. Of course, as many of you I'm sure know, we had a hurricane out here in New Haven, but in addition to that, my wife is actually about to give birth to our first child, so I was unable to miss that event. But in any case, so in case I get interrupted in the middle of the talk and have to leave, it's simply because she went into labor. Hopefully that won't happen. In any case, it's my pleasure to be speaking at this conference, although I can't attend. And let me first say how delighted I am to be speaking in a symposium with Paul Thornalley, who's really a long-term sort of idol of mine in this field of advanced glycation end products research. I'm really upset that I had to miss his talk, but hopefully at some point he and I will actually get to meet each other in the future. Let me start out by saying I have somewhat of an unusual background. I am both a synthetic chemist and a physician. And my research program, I think it's fair to say, combines those very different interests in both sort of synthetic organic chemistry and human disease biology. And what we're focusing on really is trying to leverage both of those interests by way of developing new kinds of tools for templating and manipulating complex supramolecular structures in molecular and cellular biology. Let me start by saying sort of the way that we characterize a research program is in terms of synthetic immunology. The basic idea, as I mentioned, is to use small molecules to control and or to create complex physiological functions. Human immunity is of particular interest to us. What we do specifically is to develop new synthetic methods and materials. We also then analyze the effects of those materials on proteins. So we do a lot of biophysics and interaction analysis. And finally, we like to look at we analyze the effects of our synthetic materials on cells. Why do we do it? Well, we do it for a few reasons. First off, we are interested in enhancing our understanding of the fundamental chemical basis for biology. Of course, biological systems are simply big bags of chemicals that are well sort of orchestrated to function in a certain way, both spatially and temporally, but nonetheless, they are still chemicals. And as a chemist, it is quite intriguing to start thinking about complex biological systems at the atomic and the sort of molecular level. Also, this thought process enables us, so we hope, to develop new kinds of paradigms both for disease treatment and disease diagnosis. So in that context, I'm going to be talking about two different project areas that are currently ongoing in my laboratory. The first of these has to do with a novel mechanism for treating age-related illness and other kinds of illnesses, in fact. We call this method small molecule antibody recruiting therapeutics. The basic idea is that we make bifunctional small molecules that can recruit antibodies that are already in the human bloodstream to various kinds of target cells and then induce their immune-mediated destruction. I'll talk more about that in the first part of my talk. The second area of interest has to do with the mechanism of age-related disease, in particular, the chemistry and biochemistry of advanced glycation end products. I'm supposing that you heard a fair amount about carbonyl stress and glycoxidative stress in Professor Thornalley's talk. We're interested in advanced glycation end products, also known as AGEs, from a synthetic and chemical perspective. And I'll talk more about that and some of the studies that we've been doing in the second part of my talk. So let me first start with small molecule antibody recruiting therapeutics. Basic inspiration for this project area came from the recent sort of rapid surge in popularity of antibody-based therapeutics. It's not surprising why this is the case. These materials are highly selective. They are very easy to generate against a wide variety of disease-relevant markers. And I think it's fair to say they've conventionally sort of made the refractory drug targets now druggable. They have certain limitations, of course. They need to be administered by injection. They're never orally available. They're accompanied by certain adverse reactions, including anaphylactoid reactions and immune reactions. And finally, they are somewhat costly. So antibody therapeutics range nowadays anywhere between $25,000 and upwards of $250,000 per year per patient. And so mechanisms that might enable decreases in production costs associated with antibody-type therapeutics or therapeutics that function through similar mechanisms would be highly desirable. Our complementary approach to immunotherapy involves these antibody-recruiting small molecules. We abbreviate these as ARMS. You can see these as little blue two-headed creatures right in the middle of the bottom of the slide. The way that these compounds work, as I mentioned briefly before, is they have two heads. One head binds to antibodies that are already present in the human bloodstream. It turns out that these antibodies recognize a simple small molecule epitope known as 2,4-dinitrophenyl, or DNP. By the way, I have, just for sake of making sure that my talk is, that I'm not speaking into the ether, can somebody just give me some feedback? Can everybody hear me?
Yes.
Am I on?
Yes. Can you hear us?
Okay, good. Thank you. I'll continue. So getting back to the ARMS, one side binds to these endogenous antibodies that happen to recognize a very simple small molecule hapten called DNP, or 2,4-dinitrophenyl. Not clear why we have these antibodies, although there are various hypotheses that I'd be happy to discuss at the end of the talk. But it is clear that we do have these antibodies, and they're quite widely distributed. We've looked at human serum actually from all around the United States, and we've found anti-DNP antibodies of both the IgG and IgM isotypes in every serum sample we've tested. We've now tested, I think, about 250 different samples. And so we think that this antibody population will be sufficiently robust to accommodate this sort of therapeutic, assuming it makes it that far. The other end of these bifunctional small molecules is designed to bind to surface markers for pathogenic or various disease states. And then the idea is that introduction of ARMS into a person suffering from a specific disease will lead to the formation of ternary complexes, which are perceived by the immune system simply as antibody opsonized particles that will then engage the effector responses of the immune system and lead to phagocytosis and or cytotoxicity of these unwanted species. Just by way of some of the abbreviations we use, this sort of hexagonal part of the molecule we call the antibody binding terminus, and the other sort of Pac-Man. I don't have a pointer, so it's a little difficult for me to describe some of these things. But the Pac-Man shaped pentagonal part of the bifunctional molecule we call the CBT, or cell binding terminus. There are a number of advantages associated with this approach, at least as we envision it. First off, we are employing non-toxic materials, at least materials that are designed to be completely non-toxic in the absence of a human immune system. So we're simply exploiting the built-in anti-pathogen immune response, not cytotoxic agents, which have a tendency often to have what are known as off-target effects and kill healthy cells as well. We are also utilizing self-proteins here, so the endogenous anti-DNP antibodies are just that. They're already present in the body, so they are not expected to elicit any kind of immune response. Also, of course, this strategy can be utilized against a variety of different diseases. simply by varying the identity and the recognition properties of the CBT, or cell binding terminus. From a pharmacokinetic standpoint or a medicinal chemistry standpoint, these molecules, of course, have somewhat unusual structures, but their permeability in cells is not required. And so because they do all of their sort of business on the cell surface, and so we suspect that the sort of physicochemical properties of these molecules could be quite different than conventional small molecule drugs, and they would still retain efficacy. Finally, they should be producible on very large scale at very low cost, and I can show you some of the structures in a moment, but that should enable the production and access to these molecules quite a bit more easily than protein-based therapeutics. We're not the only people to sort of work generally in this area of trying to use small molecules to manipulate immunity, and I encourage you to take a look at some work from Carlos Barbas, Laura Kiesling, Philip Lowe, Peter Schultz, and Anders Walm, all of whom really set the stage with some wonderful, wonderful work for what I'll be talking about today. In thinking about this area of research, when we started, we decided to target pathophysiologically unrelated disease states. And so we chose HIV, prostate cancer, and Staph aureus, in part because we expected that this technology could be applied to any number of disease states simply thanks to the diversity and the versatility of the human immune system. In short, in HIV, we've targeted GP120, which is, of course, the protein that mediates the very first step in virus entry into host cells. We have made a molecule called ARMH that can be used to prevent virus entry, as well as to label both virus and virus-infected cells for immune-mediated destruction. This actually has been quite successful. It's something we're now exploring in animal models for HIV. I'm not going to talk in any detail because this really is not relevant to the topic of this symposium, but I encourage you to take a look at some of our published work in the area. Prostate cancer is another disease issue that is, of course, more relevant to human aging that we have chosen to target. In particular, we focused on a protein called prostate-specific membrane antigen, abbreviated PSMA, which is expressed on the surfaces of prostate cancer cells. And we've developed a molecule called ARMP, which targets these cancer cells with picomolar affinity for immune-mediated destruction. We've published a couple papers on this, but I'll be talking about this area in more detail throughout the course of as the next step of the talk. Finally, I'm not going to talk about Staph aureus, but except for to say that we have targeted an enzyme called sortase here that has enabled us actually to feed the bacteria a small molecule synthetic substrate that causes the bacteria to incorporate DNP, an immunoenhancing functionality, into its surface. And through that mechanism, we actually anticipate that we'll be able to trick these bacteria into labeling themselves for immune-mediated recognition and clearance. I won't talk about that today, but I do encourage you to take a look at a recent paper from our lab in ACS Chemical Biology in this area if you're interested. Just to focus in a little bit on prostate cancer, prostate-specific membrane antigen is, of course, expressed on heavily overexpressed on prostate cancer cells. It's a very well-validated tumor marker. It's also expressed on the neovasculature, which supplies blood to a wide variety of tumor types, in addition to prostate cancer. Interestingly, PSMA expresses a glutamate carboxypeptidase activity. It's an enzymatic activity. And for that reason, it's been fairly straightforward to design small molecule functionality to bind and actually inhibit the enzymatic activity of PSMA. From the standpoint of our approach using these ARMs, or antibody recruiting molecules, we're not really interested in inhibiting the enzyme, but we are interested in binding the enzyme with high affinity small molecules. And so we were fortunate when we started in this project that there were a number of scientists who had already developed small molecules capable of inhibiting the enzyme and therefore binding to it with fairly high affinity. The first of these is shown here. It's called 2-PMPA. It's a phosphonate derivative, and it has a Ki of about 200 picomolar for PSMA. But we tried actually to make some ARM derivatives from 2-PMPA, and chemistry wasn't great. And so we became intrigued by this class of glutamate ureas developed by Alan Kozakowski and coworkers, which we discovered could be functionalized with various substituents. And we actually have utilized this fact to sort of inform the design of bifunctional arms targeting PSMA. To sort of further educate ourselves and inform ourselves on the likely properties of these molecules, we also did some high-level computational studies in collaboration with Professor Bill Jorgensen in our department. And what we designed on the basis of those studies was a molecule shown here called ARMP8, which stands for antibody recruiting molecule, P for prostate cancer, and 8 is the number of oxyethylene units in the linker between the ABT, or antibody binding terminus, and the CBT, or cell binding terminus, which binds PSMA. Of course, I'm a synthetic chemist. I'm not going to talk in great detail about the synthetic chemistry here, but let it suffice to say that synthesis of ARMP8 was enabled to a large extent by the late-stage application of a sharpless modified Huisgen 3+2 cycloaddition reaction, also known as a click reaction, between an azide and an alkyne. As you see here, the alkyne functionality is a part of the DNP-containing moiety labeled A, and we re-engineered the glutamate urea to contain an azide. And so access to ARMP8 in multi-gram quantities has been really straightforward for us. We're able to make, one graduate student is able to make between 500 milligrams to a gram of ARMP8 over the course of probably 3 or 4 days. Without going into all the detail of how this molecule was characterized, an important question, of course, that we wanted to ask was whether the ability of ARMP8 to form ternary complexes between antibody and target cells could in fact lead to cytotoxicity. And so what we did was an assay called an ADCC assay, or an antibody-dependent cellular cytotoxicity assay, where we combined human PBMCs, or peripheral blood mononuclear cells donated by me, to the ternary complex. And so we, in the presence of ARM, antibody, target cell, and my white cells, we measured the amount of cytotoxicity induced. And so I won't go into all the detail of this figure here, but what you see on the x-axis is the concentration of our ARM derivative. On the y-axis is the amount of cytotoxicity that we observe. And you can see the green curve here demonstrates a concentration-dependent increase in cytotoxicity in the presence of LNCAP cells, which are PSMA-expressing prostate cancer cells, PBMCs, and antibody. The other 3 curves, which do not demonstrate any cytotoxicity, represent control conditions that all together demonstrate that this cell killing is dependent upon the presence of PSMA, antibody, and the ability of our molecule to mediate ternary complexes. We have now recently taken this molecule into a mouse xenograft model for cancer. This is a humanized mouse, essentially a non-skid animal that whose immune system was partially repopulated with human peripheral blood mononuclear cells. These were not mine, and then immunized with DNP-KLH to provide an anti-DNP antibody response. And then these animals were xenografted with a human tumor line. These are the same LNCAP cells we used in our ADCC assays. And what we found was, looking at both tumor volume and survival plus and minus our ARMP, was in fact really dramatic results. And so what you see here is under a set of conditions where all of the relevant species were introduced into the tumor-bearing animals. To the brown curve at the bottom, we saw essentially stabilization of tumor volume over the course of RMP treatment. We also see a dramatic increase, significant increase in animal survival versus control conditions. You can see this red curve are the animals that have been treated with ARM and antibody. The other conditions either lack PBMCs or lack the ability to recruit antibodies. And so we are actually very excited that these molecules are efficacious in both of these endpoints. This work is actually currently under review for publication, and we're also excited to be developing this preclinically. I suspect that I'm going a little bit long because I'm sitting here in the comfort of my office and not pressured by the impatient looks of the crowd. So I'm going to skip the next section here. I apologize. Basically, just to sort of abbreviate, what we found is a sort of intriguing bell-shaped behavior of RMP as a function of concentration in terms of both ternary complex formation and the ability to induce cytotoxicity. This is actually not surprising given that the ternary complex is the active complex in RMP function. We call this phenomenon autoinhibition. We're certainly not the first people to observe autoinhibitory behavior in ternary complex forming species. In fact, such behavior is a hallmark of species that lead to ternary complex formation. It's been called the prozone effect. And because we've become interested now in this effect, we've done some mathematical modeling that actually now has allowed us, I won't go into detail just in the interest of time, but it's allowed us to model the prozone effect now analytically for the first time, which actually has eluded the community, to our knowledge, to this point. And what we've been able to do now is to go back and look in the biological and biochemical literature for so many different examples of ternary complexes mediating critical biological functions. We've now been able to reanalyze data for ternary complex forming species and determine interesting, so we hope, new insights into these various systems, including the heparin-thrombin antithrombin interaction, the human growth hormone receptor, and actually all receptor tyrosine kinase signaling processes that involve ligand-induced ternary complex formation. Of course, the MHC peptide T-cell receptor complex, other kinds of more kind of small molecule or complexes that are mediated by small molecules also are explained by our model. And so we're currently preparing a manuscript along these lines. But certainly any of you in the audience, if you have a ternary complex that you're working on that's perplexing you or striking your interest, we'd be enthusiastic to collaborate and see what analysis through our mathematical model might provide in terms of new information. Let me now talk briefly about our work in the advanced glycation end product area. And so I suspect, again, that Professor Thornalley mentioned the Maillard reaction and the ability of amines and other nucleophilic components in proteins and also, of course, nucleic acids to react with the open chain tautomers of carbohydrates and various kinds of carbonyls. And so this reaction was first characterized by Louis Camille Maillard in the early 20th century and actually is involved in the formation of aromas and flavors in foods. So you can see in this picture, Maillard reaction really has been well studied in the context of food chemistry to the extent that it gives rise to such things as the aroma of cooked rice and cotton candy flavors. And I'm certain that these chemical structures are not what most people think about when they think about cotton candy and caramel, but nonetheless, the Maillard reaction is at the core of those processes. It turns out that the human body over the course of 85 or so years, 37 degrees, and lots of glucose and other carbonyls is also undergoing the Maillard reaction, and thus proteins and other species are being glycated in vivo. We're essentially being cooked over the course of our lives by these sorts of Maillard-based reaction processes. So there are multiple pathways that lead from glucose and glucose metabolites, plus amines and other nucleophiles in the body, to a variety of really interesting structures that have been termed advanced glycation end products. At least a certain subclass of these have been termed advanced glycation end products. I show here a variety of structures that are all formed on the surfaces of proteins during the process of age formation. See here carboxymethyl and carboxyethyl lysine. These are really important modifications that are implicated in binding to certain receptors and for example, receptor for advanced glycation end product, or RAGE. Glucosapane, which is really, from a chemical standpoint, an extraordinarily fascinating molecule, which is involved in cross-linking that is thought to be really important to the pathophysiology of aging and sort of biophysical effects on collagen and other structural proteins. And finally, hydromidazolone derivatives that are shown at the bottom, which result from reactions between dicarbonyls such as methyl glyoxal and glyoxal and arginine. I venture to guess that Professor Thornalley spoke about these during his lecture. He's really a pioneer in the study of hydromidazolone derivatives and their effects, but I'll talk a little bit about our work in this area as well. Just to boil it down, our interest...
So, David...
Yes?
Yeah, you have 1 minute.
Oh! You know, I can do this in a couple of minutes. All right? I apologize. If you would, just grant me this minor indulgence, given that it's been really difficult for me to benchmark timing in my office. The one thing that I lack in here, believe it or not, is a clock. So I'll go very fast. I apologize. I really do apologize for going over. Let me just skip this slide and say that what we really want to do here is to synthesize AGEs as adducts that we can site-specifically incorporate into amino acids and therefore into peptides and proteins, and get insights in their sort of native biochemical contexts into their functions. Is it okay for me to take this additional
Okay,
time?
5 minutes.
5 minutes, okay. We're very interested in glucosapane. I'm not going to go into why. Biologically, it's very clear. This is the most common cross-link found in collagen in people over 65 years of age. This molecule, despite that it's found in all of us at varying levels, of course, has never been synthesized in chemically homogeneous form as far as we're aware. It's been isolated in very small quantities as mixtures, and we want to make it. We have actually made great progress toward this goal. I'm not going to go into detail here, but it suffice to say we're about 2 steps away from making glucosapane, and we are very enthusiastic and thankful to the SENS Foundation, who is going to be involved in the completion of this synthesis by way of a postdoctoral fellow, Dr. Christian Dragusi, who's starting in a couple of weeks. We've also managed to synthesize amino acid monomers to enable solid phase peptide synthesis of AGE peptide conjugates for carboxymethyl and carboxyethyl lysine, as well as hydromidazolone derivatives. We have a very short route to carboxymethyl lysine. This is actually a modification of a published route, as well as now really very short routes for the hydromidazolones. We can get access MGH1, 2, or 3 in diastereochemically pure form in very, very large scales and only 2 to 3 steps in very high yields. And I'd be delighted to collaborate with anybody who's interested in studying any of these compounds in chemically homogeneous form in any way that we can. First thing we've shown is that our MGH monomers can in fact be incorporated site-specifically into peptides. We've also demonstrated that although AGEs have been, of course, presumed to be just junk and sort of the mediators of damage, we were sort of wondering, might hydromidazolones be playing some other role, maybe beneficial, maybe simply physiological? And we were inspired by an observation by Professor Thornalley's lab that MGH1, the hydromidazolone, spontaneously oxidized to the corresponding imidazolone in air. And so we wanted to test whether hydromidazolones might serve as antioxidants or might be redox sensitive in vivo. We actually performed a number of assays for redox activity, one of which is the MTT assay. What we've shown here is that MGH3, in fact, is extraordinarily active in this assay. We've then gone on to look at a different assay. MTT looks at 1 and 2 electron reducing agents. The DPPH assay looks at hydrogen atom donors. And lo and behold, once again, MGH3 is about as active in this assay as vitamin C. The other MGHs are not. And so this is really exciting evidence that MGH3 seems to exhibit really potent antioxidant properties in 2 separate mechanistic paradigms. We're now doing lipid peroxidation assays to get more insight into this. But what's most intriguing from the MTT assays is that, in fact, when one performs LC-MS traces on the products of these assays, we see a new peak that's formed just for MGH3. We can go ahead now and characterize that peak here labeled B. And what we see is that versus A, the peak A, which is the starting material, the peak, the mass spec, I'm sorry, so we did mass spec, of course, high-resolution mass spec on the contents of this peak, and we see that the newly formed material now has lost 2 protons, consistent with oxidation to the imidazolone. We then went ahead and did MS-MS analyses that now are consistent with oxidation to the MGH3 imidazolone that we call MGI3. MGI3, it turns out, we were sort of thinking that MGI3 might be stable. Of course, there's evidence from Professor Thornelli's work that MGI1, or the imidazolone derived from MGH1, is stable because it's isolated and observed biochemically. MGH3, we thought maybe it's stable, maybe it's involved in mediating cross-link formation. We didn't know, so we looked at the chemical structure. It looked electrophilic. We allowed this species to incubate in the presence of nucleophile and PBS, and lo and behold, what we found is that it just simply fell off and gives rise to unmodified arginine. And so this is really intriguing to us because it may explain why MGH3 is actually not, doesn't seem to be produced in extraordinarily high forms in the human body. Maybe that's because it's being consumed. And also, this provides at least a hypothetical route for the stimulus, or redox-dependent deglycation of an advanced glycation end product. And so outside of enzymatic processes that can operate on the precursors to AGEs, and the 1-amidorase enzyme, which is present, I believe, in aspergillus. This is, I believe, the first example of a stimulus-dependent deglycation process. Although it's not enzymatic, it could actually, we hypothesized that it might be involved in post-translational regulation or pathway flux in vivo, although, of course, that's a highly speculative hypothesis at this point. I'm going to stop here.
Yeah, I think to be fair to the other speakers and the fact that the dinner hour is actually coming up here.
Yeah, you got it.
We need to end this. We really thank you for a very interesting presentation. We'll have to move on to the next speaker.
Oh, okay.
Okay, thank you.
Q&A or?
I don't think we have time. You're probably 15 minutes over.
Wow, I'm very sorry.